In this video, we are going to discuss phagocytosis, a process that is carried out by a group of white blood cells that are known as phagocytes. In the process of phagocytosis, phagocytes will bind to foreign objects, engulf them, break them down, and ultimately destroy them. The innate immune system is often said to recognize general patterns. In particular, those cells that contribute to the innate immune response recognize what we call PAMPs, or pathogen-associated molecular patterns. PAMPs can include lipopolysaccharide, peptidoglycan, and flagellin from gram-negative and gram-positive bacterium, and even double-stranded RNA from some viral pathogens. These PAMPs are recognized by receptors on the surface of these innate immune cells that are known as PRRs, or pathogen recognition receptors. Some of these pathogen recognition receptors are located in endosomes inside of the cell, but the most abundant or most well-known class of PRRs include the toll-like receptors, or TLRs. We're going to dive into detail into the process of phagocytosis, but I want to provide first a brief overview of the steps. First, the phagocyte will bind to our pathogen and engulf it to form a phagosome. This phagosome will undergo maturation and then eventually will fuse with a lysosome to produce what we call the phagolysosome. In the phagolysosome, the pathogen will be destroyed, and after this destruction, that waste will either be excreted from the cell through the process of exocytosis, or parts of the pathogen will be presented on the surface of specialized phagocytes, and this presentation will help to stimulate the adaptive immune response. Now, as we already discussed, phagocytes will bind to pathogens through the interaction between the PRR on the phagocyte and the PAMP on the pathogen. And what's going to happen is this is going to trigger an endocytosis. As that receptor pulls the pathogen inward, what we're going to find is that microtubules are going to push outward around that pathogen until that pathogen can be engulfed. And this forms then what we call the phagosome. Next, the phagosome undergoes a maturation step in which it acidifies. In this example here, in purple I am showing you the phagosome, and in green are hydrogen ion transporters that require ATP to pump hydrogen ions from the cytoplasm into this phagosome. This reduces the pH of the phagosome to somewhere between 4 to 5. This acidification step can serve as a strong antimicrobial or antiviral effect. It can disrupt proteins, which may lead to the destruction of the microorganism. Next, the phagosome fuses with the lysosome to form what we call the phagolysosome. In lysosomes, we find digestive enzymes, which will begin to break down the pathogen. These may include enzymes that break down proteins or nucleic acids. But also what we're going to find here is that within these phagolysosomes is the activation of proteins that are going to produce reactive oxygen species and reactive nitrogen species, or what are abbreviated as ROS and RNS. Examples of reactive oxygen species include hydrogen peroxide and ozone. An example of a reactive nitrogen species includes nitric oxide. Both of these will cause the oxidation of macromolecules. The oxidation of proteins and DNA will lead to not only a change in the structure of these macromolecules, but will also cause a loss in their function. For example, oxidation of DNA can lead to double-stranded DNA breaks and additional mutations. And so it is really this step here in addition to the maturation of the phagosome that leads to the destruction of pathogens within phagocytes. Now, after the organism has been destroyed within the phagolysosome, we find a couple of events may occur. One possible result will be that the debris of this microorganism is exocytosed and released from the cell. The other possibility is that if this phagocyte is what we call a professional antigen-presenting cell, then it will take parts of this microorganism and present it on the surface of the cell. This is used as a way to activate the adaptive immune response. And we'll talk about the role of professional antigen-presenting cells and their activation of B cells and T cells in later lectures.